Hello students, I extend a very warm and cordial welcome to my another lecture of Biochemistry. You are listening to Sandeepan Sengupta, who is an assistant professor in Microbiology at the Kingston College of Science. In our previous classes and lectures, we have been discussing about several aspects, biochemical aspects of protein chemistry. We came across 20 naturally occurring amino acids, the structural features of these amino acids, the very special features exemplified by each of these 20 amino acids, the forces which help to stabilize a protein structure, and of course, many more things. In my last lecture, I have explained you the biochemical and the structural features of a peptide bond which holds the amino acid residues together in a protein. Today, we will move on to the next part of our discussion, and we'll start our discussion with the biochemistry of protein structure. So, what do you mean by proteins? Hope you all know that proteins are nothing but the polymers of amino acid residues which are linked together by amide bonds or CONH peptide bonds. There are several different levels of protein structure. Broadly speaking, there are 4 levels of protein structure: primary structure, secondary structure, tertiary structure, and quaternary structure. Among these 4 levels, primary and secondary structure are considered as the 2 basic levels of protein structure, while tertiary and secondary structure of proteins are considered as higher order structure of proteins. Let us begin our discussion with primary structure. The primary structure is the simplest part of a protein structure. By primary structure, we simply refer to the amino acid sequence of a polypeptide. So what do you mean by primary structure of a protein? Primary structure is nothing but the sequence of the constituent amino acids making up a protein or a polypeptide. Next to primary structure comes a bit higher order structure, which is higher in hierarchy, which is a higher order of protein structure as compared to primary structure, and that is the secondary structure of proteins. There are several forms of secondary structures of proteins. The 2 most common forms of secondary structure, which are highly stable and are prevalent and are usually found in almost all proteins, are alpha helix and beta sheet. Now, what do you mean by secondary structure? If broadly speaking of secondary structure, we can refer to the fact that secondary structure of a protein is nothing but it refers to the local conformation of a part of the protein or polypeptide. And the 2 most stable conformations of secondary structures of proteins are alpha helix and beta sheet. Now, who were the 2 biochemists who were first to work out and come up with the structural features of these conformations? Yes, they are very well known to you. Indeed, they are Pauling and Curie, the 2 great biochemists who could work out with these two stable forms of protein secondary structures much ahead of the time, or much before of the first entire structure of a protein was elucidated. Now, at this point of our today's discussion, I would ask you to recall about the various bonds, about the 3 different bonds which make up a polypeptide. The simplest structure that a polypeptide chain could assume with rigid peptide bonds, the 2 other single bonds, that is NC alpha and CC alpha, being pre-tertiary, is a helical structure which Pauling and Curie called the alpha helix. We have discussed in detail about the 3 bonds around which rotation is expected or is likely to occur in a polypeptide. Now, I would ask you to recapitulate and recall. Hope you all know that what are those 3 bonds? The 3 bonds are the peptide with the amide linkage, the CC alpha bond, which is a single bond, and the NC alpha bond, which is again a single bond. rotation is not permitted about the CONH amide or peptide bond because it has a partial double bond character due to the presence of an electric dipole and resonance, the phenomenon exhibited by a peptide bond, about which we have already discussed in detail in our previous lecture or in our previous class. The other two bonds are the single bonds. Rotation is permitted around those two bonds, which are CC alpha, a bond called the psi bond, and the NC alpha bond called the phi bond. So how are these? What are the structural and conformational features of the amino acid residues present in an alpha helix, and how are they arranged? Actually, the amino acid residues are arranged in such a way so that the length of the helix extends about 5.4 angstrom along the longitudinal axis. Thus, an alpha helix basically extends for 5.4 angstrom and consists, each turn of an alpha helix is made up of 3.6 amino acid residues. The amino acid residues in alpha helix have conformations with CC alpha bond, that is psi, lying between -45 to -50 degree, and NC alpha bond, that is phi, lying between or lying around 60 degree. And each that is segment of alpha helix consists of 3.6 amino acid residues. The twist of alpha helix, found in almost all proteins, is predominantly right-handed. And another interesting feature is that alpha helix has been found to be the most prevalent and predominant structure found in alpha keratids. Now, if we take a look over the alpha helix or over the secondary structure of almost all proteins and polypeptides, a very interesting feature which we'll come across is that almost 1/4 of the amino acid residues present in every protein or polypeptide adopts a secondary conformation called alpha helix. So, alpha helix is in fact the most stable conformation among all different forms of secondary structure of a polypeptide. So, what might be the reason behind this? The reason behind this, the most prominent reason behind this, is all about hydrogen bonds. The answer lies in part that an alpha helix makes optimal use of internal hydrogen bonds. Now, we came across several structural features of alpha helix. As a part of your work, I would ask you to go through the biochemical features of a hydrogen bond. What are the different kinds of internal, intra and interhydrogen bonds which are present in proteins or which help to stabilize a protein structure, because we have already studied in detail about the forces stabilizing polypeptides or proteins. So, as a part of your reading, you can study more about the structural features of alpha helix from Lehninger principles of biochemistry, and meanwhile study about the special features of hydrogen bonds and about how the structural features and the biochemistry, how hydrogen bonds are produced and how hydrogen bonds help to stabilize, or internal hydrogen bonds stabilize the structure of alpha helix. So to summarize, the important structural features of alpha helix are: number one, it makes optimal use of internal hydrogen bonds. Number two, the amino acid residues are arranged to form 5.4 angstrom along longitudinal axis. Each includes 3.6 amino acid residues. The helical twist of all amino acid residues present in alpha helix is right-handed, and of course alpha helix is predominantly the most stable of different secondary forms of secondary structure found in almost all proteins and polypeptides. So keep studying more about the hydrogen bonds. In the next part of my today's talk, I'll be coming into the detail of the internal hydrogen bonds which help to stabilize the alpha helix, making it predominantly the most stable form of secondary conformation found in almost all proteins and polypeptides. So till then, stay tuned for the next part. I'm resuming back after a short break, and till then you can study more about hydrogen bonds and about the back features of alpha helix through Leninger principles of biochemistry. Stay tuned. Goodbye till then.